Welcome, everyone. Thank you for being here at our Friday noon seminar. Today, we have the pleasure of introducing Dr. Rikki Lissaman. He's a postdoctoral fellow based at the Douglas Research Center at McGill University, originally from the UK. He has a BSc in psychology at the University of Birmingham, an MSc in brain imaging at the University of Nottingham, and a PhD in cognitive neuroscience at Cardiff. So today he's going to be talking to us about using MRI to investigate things that are not maybe fully typical in a physics talk, menopause and sex-related differences, and episodic memory. You're the expert, I'll let you talk. Thank you so much.
Thanks so much for the introduction and also for the invite to come and give a talk outside of my main discipline. So as mentioned, today's talk is going to be a little bit different, maybe a lot different to what you're used to seeing. Rather than focusing on methods development, I'm going to talk a little bit about how we, that is cognitive neuroscientists, use these methods to study the relationship between the brain and cognition. And in particular, I'm going to talk about how our lab uses MRI to study the neural correlates of episodic memory at midlife and the role of sex and menopause in that regard. So, there we go. Okay, so I'm going to start with the most basic question: what is episodic memory? So the term episodic memory refers to the encoding, storage, and retrieval of episodes or events situated in a particular time and space. So you can think about this as kind of what, where, and when memory. So an example of this might be a memory for what you did on your last birthday. Perhaps it's a camping trip with friends or something like that. So it's situated in both time and in place. And the term was originally coined in 1972 by N. Tulvig in an attempt to distinguish episodic memory from semantic memory, the latter of which refers to memory for general concepts, facts, and knowledge about the world. So both forms of explicit memory, memory we can talk about, bring to mind, but subtly different in its content. And one of the important aspects of episodic memory is this is context. So context plays an incredibly important part of episodic memory, and it's widely used today to study the phenomenon. So in terms of the actual paradigms that we use to study episodic memory, one of the most common is source memory or context memory paradigms. So that just reiterates the point about context being really important in episodic memory. So I'm going to give you a very, very brief overview of what this looks like. So usually this involves an encoding phase, a break or a kind of storage or consolidation phase, and then a retrieval phase. So during the encoding phase, participants are usually shown a series of stimuli. Now here it's just a cartoon of a lion, but it could be a picture of a scene, it could be a video, it could be anything. But it usually involves some form of manipulation about where on the screen it's presented or when it's presented. So if you were using words, for example, you could have list A, list B, list C. So the important point is that we're not just presenting items, but we're also manipulating where and when. Then after a short break, usually on the order of minutes, so I'll come back to that. But then in terms of retrieval, we then show some of these items again, as well as some new, and we ask the participants to make a judgment. We ask them to say whether or not they've seen these items before, but importantly, where they saw it on the screen or when. So by operationalizing episodic memories in this way, we can get at the what, where, and when of episodic memory in the lab, or more relevant to this particular talk, in the MR scanner. Now, there are various different types of, there are various different ways that we can use MR to study the neural correlates of episodic memory. One of the most common is fMRI, or functional magnetic resonance imaging. Now, when I was putting together this talk, I debated how in depth to go here, considering I am a cognitive neuroscientist, and this is a room of medical physicists, and it kind of feels like having a hockey player tell people on the house, for example, how to play hockey. So I'm just going to give a very brief overview for anyone who might not be familiar with the technique, but detail is going to be a little lacking. So in terms of at rest, neurons require oxygen as part of normal respiration, and this is delivered to them by hemoglobin in capillary red blood cells. Now, when a neuron is active, so when it sees a stimulus that it responds to or is sensitive to, activity increases, and that means that there's an increased demand for oxygen. Now, the local response is an increase in cerebral blood flow, which brings with it an increase in oxyhemoglobin and a relative decrease in deoxyhemoglobin. And that is important for us because oxyhemoglobin oxyhemoglobin and deoxyhemoglobin have different magnetic properties. Oxyhemoglobin is diamagnetic and deoxyhemoglobin is paramagnetic. And because paramagnetic substances distort the surrounding magnetic field, nearby protons will experience different magnetic fields and will start to precess at different frequencies. So that means that the presence of deoxyhemoglobin results in a loss of signal. So putting that together, because we see an increase in the ratio of oxy to deoxyhemoglobin, we see an increase in the signal. And that underpins much of the blood oxygen level dependent signal that underpins most of the work that we do in fMRI research. In terms of how cognitive neuroscience is actually applied this, so using those kind of paradigms that I showed you earlier, we study how brain activity changes while people conduct these tasks in the scanner. And many hundreds of studies have been conducted on this topic. And what they tend to highlight is that a particular network of regions are active when people are recalling episodic information. And these regions include the hippocampus, which is part of the medial temporal lobe. It includes part of the prefrontal cortex, the medial aspect. And it also includes posterior cortical regions such as retroscenial cortex and posterior cingulate cortex. And this maps on quite nicely as well to what we know about things like amnesia patients and where their damage tends to be. Well, fMRI isn't the only game in town. There are other forms of MRI that we can use to study the neural correlates of episodic memory. So this includes resting state fMRI, structural MRI, and diffusion MRI. So resting state fMRI, like task fMRI, relies on the bold signal, but instead of looking at changes in activity while participants perform a task, typically participants lie in a scanner, either looking at across or having their eyes closed, and then we measure the temporal synchrony in the bold signal across regions. And that synchrony is sometimes referred to as intrinsic functional connectivity. When it comes to structural MRI, this typically relies on T1 or T2 weighted scans, and from that we can derive neuroanatomical properties such as grey matter volume, cortical thickness, things like that. And then there's diffusion MRI, which we can use to study white matter microstructure, and I'll return to that a little bit later on. But the key thing is that all three of these methods give us metrics that we can correlate with performance on a task, which kind of helps us get at which regions and which white matter tracks might have a role in episodic memory. Now, that's a little bit about what episodic memory is, how we study it, and in particular how we study it in the scanner, but why should we care? Well, for someone like me who's interested in an aging brain, episodic memory is particularly interesting because it's so vulnerable to the effects of age. And that's true both of healthy or typical aging as well as pathological aging. So in Alzheimer's disease, for example, one of the earliest symptoms you will see and one of the most marked is a deficit in episodic memory. So this highlights just how important episodic memory is from across the lifespan and also for our kind of quality of life. Yet despite that, we currently have no interventions, whether pharmacological or lifestyle, that are capable of ensuring that we maintain good memory into late life. So to potentially prevent or delay Alzheimer's disease. And what we need to know for the development of those sort of interventions, among many things, is two key things. When the decline begins, so that we know when to intervene, but also what factors exacerbate or protect against this decline, because that will give us not only more of a mechanistic understanding about what's happening with the decline, but it will also help us understand who will benefit from those kind of interventions. Now, as I mentioned, we know from both cross-sectional and longitudinal research that episodic memory declines across the lifespan, but this research tends to focus either on older adults or on comparisons between older adults and younger adults. And that means that we tend to miss out on midlife. Yet, our lab and others have started to show in recent years that there are already changes in memory for spatial context, that's the where of episodic memory, at midlife, and that this is related to variation in both the structure and function of the brain, so particularly impacting things like the hippocampus and the prefrontal cortex. So what seems to be emerging from our research is that there's a real importance of midlife. And this also works quite nicely with things like the Alzheimer's literature, where there's an increasing appreciation for the fact that Alzheimer's pathology often comes many years, perhaps even decades before clinical symptoms start. So this underscores the need for why we need to look at midlife. But midlife is also important for another reason, and that's because it is a time in which females with ovaries, so people who have undergone an oophorectomy, for example, will transition from pre to post menopause. So for anyone who might not know, menopause refers to the permanent cessation of menstrual cycles due to the loss of ovarian follicular activity, and also is associated with a decline in blood estrogen levels. Now, this typically happens between ages 45 and 55, so in the middle of that midlife period, with an average age of onset around 51 or 52. And while experiences of menopause vary, with some females experiencing relatively few symptoms, others experience quite a lot, some of which are shown on the screen here. And well, some of the spacing's got a bit strange, but it's all good. So you can see how the experience of menopause varies quite dramatically. But from our perspective, one of the most interesting things is that many females self-report issues with cognition and in particular memory. And in the literature, this is referred to as kind of brain fog or menopause fog. So this led our lab to kind of ask the question, well, we're seeing midlife effects on memory and its neural correlates. Maybe that's being driven by postmenopausal females or people who are going through the menopausal transition, and maybe that's contributing to sex differences. And that then adds into this idea about who would benefit from kind of our understanding of memory across the lifespan and potential future interventions. And there's good biological reason to believe to think that there might be some signal here. And that's primarily from both research on rodents and non-human primates, which has shown that regions such as the hippocampus and prefrontal cortex, which we know are important in memory, are very sensitive to estrogens and estradiol in particular, and impact kind of animal equivalents of the tasks we would think might tap into episodic memory. So from this, we can't make a simple leap from animal models to humans, but it does kind of suggest that there is a plausible mechanism at play here for why there might be menopause and in turn sex effects at midlife. Now, despite this, and despite the many studies that have been conducted using various forms of imaging and what might be referred to as the cognitive neuroscience imaging, reproductive aging or menopause tends to be neglected. So here you can see a figure from Taylor et al. Who conducted a review on cognitive neuroscience of aging papers published between 1995 and 2017. So this is papers using various forms of MRI, EEG, all sorts of different methods. And what they found is that despite a massive increase in the number of papers looking at chronological age, there are comparatively few looking at reproductive aging. So these yellow bars down here that you might not be able to see, the studies that actually factor in these differences, these effects that specific to females. But it actually gets worse than this, because among those yellow bars are studies that don't include things like, they include blood-based hormone measures, so that the staging of females according to where they are in their menopausal transition is probably inaccurate. So all of this is to say that while we have the tools to study it, i.e. MRI, very few people are actively looking at this. And so we have a huge gap in our understanding of the aging brain, and particularly what happens in midlife in females. So this is where our lab comes in. So over the last 2 or 3 years, we've been recruiting and testing participants from across the greater Montreal area. And one of the main goals of this study is to understand exactly that. So the neural correlates of episodic memory midlife, but also to understand whether or not menopause is having an effect on them and in turn contributing to sex differences. So I'll give you a brief overview of what the brain health and memory at midlife study actually comprises. So it's a 3-stage kind of study. So at enrollment, we do all the standard things you would expect: consent forms, questionnaires to make sure our participants don't have severe neurological illness, severe psychiatric illness, that kind of thing. And if they pass our inclusion-exclusion criteria, they come into the Douglas Cerebral Imaging Center, where we perform a number of neuropsychological assessments. So that's things like the mini mental state exam, the Beck depression inventory, all of these kind of things, so we know where they're at in terms of their functioning. But they also complete questionnaires where they tell, particularly the female participants will tell us about their reproductive histories, so whether or not they're still cycling, whether or not it's stopped, if they have any conditions like PCOS and things like that. And then if they are good, we bring them back for a second session at the imaging center where we conduct MRI scanning. And we use a number of different sequences to collect different types of images. So we collect T1 and T2 weighted images for structural imaging. We collect resting state fMRI. But the two for this talk that I'm going to mention, and the two that I'm going to focus on, I should say, is task fMRI and diffusion weighted imaging. There we go. So the first project I'm going to talk about is where we use task fMRI. So this comes from a paper that we recently published in the Journal of Neuroscience. It was led by a former master's student in the lab, Ariel Pestol, who's now based in Norway. And in this project, we asked 2 main questions. So we were basically interested in the effect of menopause on the association between kind of episodic memories tested using these spatial context tasks and brain function encoding and retrieval using fMRI. But we were also interested in how chronological age would be related to this within each group. And we were asking this question because menopause is an age-related process. So we would expect age differences between people who are premenopausal and those who are postmenopausal. But it's possible that the effect of age within those groups differs. So we wanted to kind of break this down a little bit further. The sample itself came from the overall BHAM study. We focused on the middle-aged females with ovaries, so we didn't include males in this study. And we split everyone according to pre and post menopause states. So we took their reproductive histories, we took their blood-based hormone measures, and we were able to stage them using the straw 10 criteria, which allows us to be relatively accurate in placing them as to whether they were pre or post. As expected, we get differences in terms of chronological age between these two, which you can see on the screen. Both are definitely within kind of what we would classify as midlife, which is usually considered 40 to 60, but if you're being a little bit more generous, you might stretch that up to 65. But we get the expected difference where premenopausal females are younger than the postmenopausal females, and we also get the expected differences in hormones. So, for example, estradiol was lower in post than pre, and FSH was higher in post than pre. Now, in terms of the actual imaging that we use for the task fMRI, so I've provided a brief overview here. So over at the Douglas, we have a Siemens 3T Prisma Fit scanner that we use, and for this particular imaging sequence, it's a single-shot T2 star weighted gradient EPI pulse sequence. And I've included the parameters on here. I would argue, as a non-methods person, this is pretty standard for whole brain task stuff. Obviously, with say 70, or if you take an ROI approach, you could increase the in-plane resolution and things like that. But because we're interested in the whole brain, that's reflected in the parameters here. And in terms of the actual design, so how we measured episodic memory while people are being scanned, you can see that this is very similar to the cartoon that I showed a little earlier, but with a few key differences. So the actual stimuli here are black and white faces, the aging variant, so it includes, excuse me, it includes toddlers all the way up through to older adults, so across the lifespan we get a range. And those images are presented in one of 4 quadrants, so top left, top right, bottom left, bottom right. And participants are just asked to make a judgment about whether the face is pleasant or neutral. So they don't say unpleasant, just pleasant, neutral. And the whole point about that judgment is not that we're measuring it, it's just to make sure that they're paying attention on the screen, they're looking and actually actively encoding those items. Then later on during retrieval, they're shown some faces that are old and some faces that are new, and they're asked to respond accordingly. So they're asked to say that it was whether or not it was familiar. So this is the F down here, where they say, I've seen it, but I couldn't tell you where on the screen it was. Or they're asked to give the location, so they can say, yeah, I knew that I saw it and I knew where it was, or to say that it's a new item, so to try and project it if it's an old one. And we asked them how well they thought they did in the intervening periods, the breaks. And importantly, they're scanned during both encoding and retrieval, I should mention that. So neither of these are out of scan. In terms of the participant responses, we can categorize how they respond on this task using signal detection theory. So here on your right-hand side, you can see the correct responses. So whether or not the item was new or whether or not it was old and at a particular location. And there's 6 possible responses that the participant can give. So if you just focus on if the item is in the top left, you can see here that if they say it was new, it's a miss. If they say it was familiar, but they couldn't remember the exact location, we call that recognition. And then we have source misattribution for if they remember it but get the location wrong, or source correct if they remember it and get the location correct. And what we're interested in primarily is those source correct responses. So we refer to this as spatial context retrieval accuracy. But basically what we're doing is taking all of those source corrects and we're dividing by the number of old items that they saw. So this is just a measure of how well participants were able not just to remember the items, but also where on the screen. So we're tapping into that contextual component of episodic memory. Now, in terms of analysis, you'll find a lot of labs at this point will do kind of like a standard univariate analysis. We take a slightly different approach. So we use a multivariate statistical technique referred to as partial least squares. And in particular, we tend to use behavior partial least squares, which falls under the kind of partial least squares correlation family, which is basically just analyzing it's analyzing the information common to both brain on the one hand and behavior on the other. So I'll just give a very brief overview of what this is actually doing. So here you can kind of see that we start with 2 matrices. So in X, we have brain activity. So each row is participant, each column is activity in a voxel at a particular time. So we compress time and space over the columns. And then in the Y matrix, we include behavior. So this is things like the spatial context and memory performance that I was mentioning earlier. Now, within these matrices, you can have sub-matrices corresponding to conditions. So we have an easy or hard task manipulation, so you can incorporate that. But you can also incorporate groups, so pre and post menopause, for example. Now, at this point, we cross-correlate them to give us this new matrix R before then applying singular value decomposition. And this gives us orthogonal latent variables that maximally explain the covariation between these original matrices. And we test the significance of these using non-parametric permutation testing. And for any significant latent variable, we tend to then go on to look at singular value. So this represents the amount of the crosswalk covariance that's accounted for by the latent variable. The singular image, which shows positive or negative saliences or weights that basically capture the voxel contributions to the overall pattern. And that pattern is expressed by the correlation profile. So this shows how the behavioral measures that we included at the start are related to the patterns of brain activity that we see. So in terms of what we actually found in the task fMRI project, so this is purely behavior or cognitive performance. So I didn't go into necessarily about how we analyze this data, but the top part is with partial least squares regression, and the bottom is with linear mixed effects models. I won't go into much more detail because I want to get into the imaging stuff. But essentially what we found here is at the top, you can see that postmenopause status in advanced age was associated with poorer spatial context memory. So you can see how the bars are moving in different directions here. So basically, the older a participant was, or if they were postmenopause, they were less likely to be able to remember where the item, what the item was, and where it was presented. Now, we also wanted to have a look at age within groups, and this is where it got a little bit interesting, because within the postmenopausal females, we saw much stronger relationships with age than in the premenopausal group, which we think might be related to the fact that there's kind of like a sort of interaction effect where age effects become more prominent after menopause than before menopause. As though something happens in the transition from pre to post is increasing vulnerability to age effects. In terms of the actual imaging work, though, so this is the output from the partial least squares analysis. So we identified two statistically significant latent variables. This is the first one, which I propose as LB1 here, and it accounts for roughly 15% of the covariance in the original matrices. Now, I'm going to try and break this down a little bit because it's a little confusing. But what you can see here in the singular image is the positive saliences indicated by warm colors. So these are the voxels that were related to the pattern shown in the correlation profile. And these are primarily in regions of lateral prefrontal cortex, and the gyrus inferior parietal cortex, and midlife cortical regions such as posterior singular cortex. So these overlap with the sort of regions that we know are involved in episodic memory and that I showed a little earlier. But in terms of the actual pattern of correlation, so we can see that the patterns are more strongly related to premenopausal females than postmenopausal females here, with increased activity in these regions related to spatial context memory retrieval. So that's this middle part here on the left-hand side. So the more activity in these regions, the better participants did in recalling not just the item, but also where it was presented on the screen. And that was true of both encoding and retrieval, which is interesting for us because that kind of ties in with this concept of reinstatement, the idea that regions that are active when we encode an item are then reactivated when we retrieve that item from our memory, which suggests that participants are relying on this network of regions at both encoding and retrieval when they're then asked to kind of perform the task. But what we also found here is some interesting, a slightly interesting relationship in the postmenopausal group, which is that age was positively correlated. So the older the postmenopausal females were, the more likely they were to increase activation in these regions, but it had a negative relationship with performance. So to try and interpret this, the way that we think that this is happening is that, or what we think is happening, I should say, is that we think that the older the postmenopausal females are, the more they're trying to tap into these regions that younger adults rely on in order to compensate the kind of neural inefficiencies. And that's why we're seeing a detriment in behavior despite activation in these regions. But we also identified another pattern of results from this analysis in which it was more closely related to the postmenopausal females than the premenopausal females. So again, you can see here that this latent variable accounts for roughly 10% of the crosswalk covariance. And it was mainly associated with negative salience regions. So that's indicated here by these cool colors, which were located primarily in ventral occipital temporal cortex and parahippocampal gyres. So different regions to what we saw in the previous latent variable. Now, one thing to note, when it's a negative salience, these relationships are flipped. So if it's a negative bar, it's so instead of being increased activation being positively related with anything going up, it's actually negative related with going up. So that's important interpretation. So what we're actually seeing here, if you focus on the spatial context in the middle for the postmenopausal females, activity within these regions, increased activity in these regions, both on the easy and hard version and encoding and retrieval, was associated with better spatial context memory. So it seems that the postmenopausal females were relying on different regions for this test than the premenopausal. And interestingly, we also found that with age, they were less likely to activate these regions, potentially suggesting that there's an age-related decline in the ability to kind of activate these regions, which supports performance. So that's kind of related to the performance-related decline that you might see. Now, that's quite a complex interpretation of our results, but the key thing here is that at both the cognitive level and at the imaging level, what we were seeing in this project is menopause-related differences in the direction of age and performance-related correlations with that activity. So it does seem like at midlife, there are menopause-related effects that are of relevance to episodic memory. But I did mention earlier that we collect other types of MR images, and the one that I want to talk about here is diffusion-weighted imaging, or DWI, sometimes referred to as diffusion MRI. So again, I don't necessarily want to go into this in too much detail. Diffusion-weighted imaging essentially involves the application of gradients that cancel out when water molecules are relatively stationary, but when they're moving, what you'll see is you'll see dephasing and a loss of signal. So in diffusion imaging, we exploit signal loss. And this is important here because of this biological concept of restriction or hindrance. So you can see here on this cartoon roughly 3 tissue types. So you can see CSF, gray matter, and white matter. Now in the CSF, so all of these shown here are kind of random walks of water molecules. Now in the CSF, what you can see is that that water molecule can essentially move in any direction unhindered, basically. And in such cases, it's relatively free or sometimes with isotropic diffusion. So if we use diffusion MR, we apply gradients in different directions, but in CSF, regardless of that direction, it's going to essentially appear as though diffusion is high, it's going to appear dark on those scans. With grey matter, it is more restricted or hindered, but not in any particular direction. So again, so diffusion is going to be a little lower here, so it's going to appear a bit brighter than in the ventricles. But in white matter, this is where it gets interesting. So the presence of axons, it imposes direction on those water molecules and undergoing random walk. So this is what we refer to as anisotropic diffusion. And that's useful for us because it helps us to have a contrast that we can pick up on white matter in particular. So here are some examples of diffusion images with just 3 different directions, gradients applying 3 different directions, I should say. So left, right in A, anterior, posterior in B, and superior, inferior in C. So as I mentioned, in the CSF, you can see how it appears dark because diffusion is high in pretty much all directions. But I want to focus on where the arrow is pointing here. So hopefully you can all see this little white arrow. It's not the clearest, but hopefully you can see it. So here what we can see is that when there's a left-right, once a gradient applied from left to right, this area that the arrow is pointing to appears dark. When we apply a gradient anterior-posterior, it appears quite bright, particularly in the middle point. And then when we apply it superior-inferior, it appears bright across the whole kind of region that it's pointing to. So what we can infer from this already, from just 3 scans, 3 different directions in which gradients are applied, we can already work out that the white matter that we're looking at here probably runs from left to right. And that is consistent with the known anatomy of the corpus callosum, which is shown down here. So you can already start to see how we can get information just from a qualitative perspective about the white matter from this. But that doesn't really tell us much about, well, it doesn't give us anything that we can use to analyze quantitatively. We can't correlate any of this with episodic memory. So the question is, how can we model the degree of anisotropy in the white matter? This is where the diffusion tensor model comes in. So it was a particularly early model that was proposed, but has remained extremely popular. And again, I'm not going to go too much into detail here today, but basically the idea here is that you can see here on the left that there are axons imposing directional dependence on water molecules that may be undergoing diffusion, and that we can capture this mathematically and also visualize it as an ellipsoid, which gives us a way of generating a number of quantitative metrics about the way in which water molecules are displaced in a given voxel. And that includes, so the two of the most common metrics that we derive from this are fractional anisotropy, or FA, and that represents the degree to which diffusion is constrained in a given direction. So this would be an example of quite high FA. So it's from 0 to 1, so this might be something like 0.8, for example. Then you have mean diffusivity, or MD, which represents the average mobility of water molecules in a given voxel. And FA and MD are usually anti-correlated. So if you have areas of high FA, they're usually associated with areas of low MD and vice versa. But what's particularly important for us is that from animal models and various other forms of evidence, we know that these metrics are sensitive to the microstructural properties of white matter. So things like axonal cell membranes, the myelin, and various other kind of macromolecules that may be present, but they're not specific to any one. So we can't use these metrics to say, yes, there's a problem with myelin in this person, or the myelin is related to this particular aspect of cognition, but we can say something about the way in which microstructure as a whole in the white matter might be related to cognition. Now, taking a step back, why do we care about white matter in this context? Well, there have been numerous studies published showing age and sex effects on the microstructural properties of white matter using both diffusion tensor imaging and other methods. And some of these reports indicate that age effects on white matter are more pronounced in females, which has been hypothesized to be linked to menopause. So this immediately makes it a relevant topic for our research. And again, there's biological reason to believe this might be the case. So from animal research again, the cognitive effects of estrogens, particularly estradiol, have been linked to direct effects on neurons, which result in synapse loss and lower connectivity. Now, because the white matter tracts are the brain's key structural connections, this bridges the gap between the two and suggests that we might be able to detect something similar in humans. So as part of this project, we kind of answered two related questions building on the fMRI project. So we were interested in whether or not menopause affects the association between age and white matter microstructure. So because the literature has shown that there are age and sex effects, for example, we wanted to know more about that. But in turn, we wanted to know how that might be related to spatial context memory. And this is a paper that we've currently got in review. So the sample, again, came from the overall BHAM study, but we incorporated males this time, so we could get at the sex differences effects. We could get a look at sex differences and if they were present. The females were again split into pre-post-menopause, and it's worth pointing out that the overlap in samples is high but not complete. So some participants, they didn't complete the diffusions imaging, but they did complete MRI and vice versa, that kind of thing. We found sex differences in hormones, as you would expect, and again, given the overlap in sample, menopause-related differences in age and hormones. And I just want to point out, just to make it absolutely clear, we ran two analyses here. So we did a menopause analysis, so we compared the pre to the post, and we also ran a sex analysis where we collapsed across menopause status and compared against the males. And just to make this especially clear, we did some age matching as well and found that if we reduce the number to make it more equal across groups, it really doesn't affect much of the outcome. So again, here's more about the acquisition protocol. So the only thing I really want to flag here is that we used a kind of a single shell acquisition approach. So we only applied one diffusion weighting in scanner. So we weren't able to do any of the more fancier things that happen in kind of diffusion imaging. So we weren't able to kind of look at intracellular and extracellular water components, for example. But this is pretty standard from like a clinical perspective. You could get this sort of thing in clinic for sure. And we also collected additional non-diffusion weighted volumes in the reverse phase encoding direction for susceptibility to distortion correction. And if anyone's interested in pre-processing, I can talk about that a little after. In terms of the actual analysis, so we fit these diffusion tensor models to each voxel, which gave us these kind of FA images or MD images. And then at that point, we applied what's referred to as track-based spatial statistics. So that's where we take the FA images, we kind of merge them all, well, we register them all into common space, so MNI template in this case. We generate an average image and then we skeletonize. So we're able to identify regions in which white matter is common to all of the participants. We then project the FA and MD values onto that skeleton, before then registering it to this white matter atlas. So we were able to generate mean FA and mean MD values for each of these ROIs. And you'll note that if you're familiar with this atlas, that we didn't include some of the brainstem ROIs because of limited coverage in some participants. And we also collapsed across left and right hemispheres where relevant because we didn't have an a priori hypothesis about hemispheric effects. And again, we used partial least squares for the association between age and white matter microstructure, but we just adjusted it. So instead of performance, we had age in the Y matrices, and we had microstructural measures in the X matrix. So in terms of the results, starting again with cognition first, so this is just behavior. What we found here is a very similar pattern as you would expect to the first project. So post-menopause status and age, both related to poorer spatial context retrieval accuracy, with stronger age effects within the post-menopause group. But where we were able to extend this further is that by including a sex comparison, we showed that the relationship between age and performance appears relatively specific to females. So this age effect in post-menopause seems to be driving a decrease in performance across midlife that is not present in males. So it doesn't seem to be the case that this is quote unquote just an age effect, which is something that we like to tell reviewers. In terms of the imaging itself, so this is again takes a slightly different approach to the what I showed earlier because there's no behavioral measure in the Y matrix. Instead, what you've got is a correlation profile showing how age was related to FA and MD. So again, we identified one significant latent variable in this analysis, which was associated with negative saliences in the anterior coronal radiata and the body ingenue of the corpus callosum. So that's shown up here in the singular image. And what this correlation profile essentially shows is that in both premenopausal females and postmenopausal females, lower values of FA and higher values of MD were associated with advanced age. And that's because remember, it's negative salience, so that relationship is flipped. So I should have, if I didn't mention earlier, high values of FA and low values of MD are usually interpreted as healthier microstructure, higher structural quote-unquote integrity. And so what this is showing us is that there's a decline in that kind of microstructural health with advanced age, irrespective of whether or not somebody is pre or post-menopause. But PLS allows us to do something a little bit extra, which is that it gives us these what we call brain scores. So these brain scores shown here represent the degree to which an individual participant exhibits this pattern. So the higher the value of the brain scores, the more representative this pattern is of their microstructural properties. And when we related those brain scores to cognitive performance, what we found is that the more that participants show, the more that the postmenopausal females showed this age-related decline in white matter microstructure, the poorer they did with spatial context. But that wasn't the case for premenopausal females. So that comes back to this idea that I mentioned a little earlier, that maybe there's something about the transition from pre to post-menopause that increases vulnerability to age effects. And this time we're seeing that on both the brain and cognition link. For the sex analysis, which we conducted essentially in the same way, but just with slightly different groups, we find a very similar pattern with the latent variables. So the anterior coronal radiata and the genome of the corpus callosum were implicated, with the fornix also. And we get the same sort of pattern. So FA is lower values of FA associated with advanced age and higher values of MD associated with advanced age. So poorer microstructure as we get older. But when we map that onto cognition, although they show the same pattern, females were the only ones that actually showed a significant correlation there. So from that, it's weaker than in the menopause analysis, but we think that that might be related to the fact that postmenopausal females are driving this. And actually the premenopausal females who show a much weaker relationship are making this look more moderate, basically. So this suggests that maybe the menopause effects are contributing to sex differences at midlife. And this is just briefly summarizing that. So at behavior, we see that both postmenopausal status and advanced age are related to poorer spatial context memory. But in this project, we were also able to show that that might contribute to sex differences in age trajectories. We also showed that age at midlife is associated with quote-unquote poorer microstructure, predominantly in frontal white matter. And this is important because there's this idea in the literature that age effects have an anterior-posterior gradient. So anterior or frontal white matter tracts are more affected by age and affected earlier in the lifespan than more posterior white matter. And this is consistent with that idea. And also that the expression of this decline is related to memory in females and postmenopausal females in particular. So kind of putting this all together, just very briefly, using MRI, a non-invasive imaging method and its variations, we were able to observe evidence that the transition from pre to post-menopause increases vulnerability to age effects. And that's evident both at the cognitive or behavioral level and the relationship between cognition and the brain. And that's true of both function and also structural connectivity, the white matter microstructure. So if we zoom out even further, what I think I want to take home for everyone to be really is that not only is imaging useful for studying the relationship between the brain and cognition, but also that as we want to understand the aging brain and how cognition and brain both change with age, we really need to be paying more attention to these kind of menopause and reproductive aging effects, because it might be the case that we're missing out on important pieces of the puzzle, and that future interventions might rely on our understanding of these effects, and also influencing who might benefit from potential treatments. So at that point, I'm just going to say thank you to everyone who's been involved in this work. So all of the Raja lab, so that's our PI, Natasha Raja, and all of the various people who've been involved in collecting data, analyzing data. You know, as a postdoc, I'm very fortunate in the sense that I don't have to do a lot of my own data collection anymore. And these are the people who actually do the work. And yes, I also want to thank our funders. So CIHR provide a lot of funding for this project, also NSS as well. And with that, if anyone has any questions, I'd be more than happy to answer them.
All right, thank you very much. Excellent talk. We're open for business, open for questions. So in the room, please ask questions. If you're on Zoom, you can either unmute yourself and ask, or if you type it in the chat.
And I appreciate that it's quite a different talk to what you're used to. So if the you know, if questions that immediately jump to you, I understand why. But
It's a question online. So raise them.
Who raised your hand? Nita, please go ahead.
Yeah, well, thank you so much. This was a very interesting talk, and very different. So I do appreciate that as a part of the MPU. So I have a twofold question. I hope that's okay. One is very simple. So how about hidden variables? You know, you stratify women based on age, you know, pre and post menopause, but how about quality of life, some cognitive activity there, diet or exercise? Obviously, you know, these things could affect neurocognitive performance. And then my second question is, I'm a little concerned as a woman when we use listen, I'm serious about this. When we use these terms as neural inefficiencies, cognitive decline, you know, women have gone through a lot of hassle just to be educated and earn, you know, own property, and things have been traditionally considered as not able to do so with the same level of success as men. So I'm a little concerned that this could, if we take this seriously, you know, at the face value, we could affect, you know, potentially hiring women at that age and saying, well, you have so much brain fog that you cannot really be the CEO of this company. So anyways, that's my second question. I know the second is a little more sensitive, but it's my honest question to you.
that's fair enough. So can you just repeat the first question? The second question.
Yeah. How about, like, I think your plot where you compare, you have male and female. I'm not sure that was the white matter one, a diffusion method, and then you really show that versus male with the blue line with a band was pretty straight, and there is a slope with age for females. I mean, that slope obviously seems significant. There is some p-value quoted there. But how about if you stratify that sample for different, as I said, I don't know, you know, different levels of education, quality of life, and diet, different things that affect cognitive performance within that group of women. So that's my first question, yeah.
Sure. So taking the first question, so we stratify based on menopause, so where the females are in their kind of menopausal transition rather than age per se. Like it is age related, as I said, but we mainly use the information they provide to us in questionnaire form about their reproductive histories, but also about blood hormone levels and things like that. Now, you are absolutely right though that there are other variables that are at play there. So also I mentioned on the screen, I showed some commonly reported kind of symptoms. It sounds too clinical, but symptoms of menopause where some people report difficulty sleep, and it's possible that that might be related to the cognitive performance that we're seeing. So I absolutely agree that there are other variables that might be relevant here. And that's something that we're slowly going to start looking at over time. So we're interested not just in, say, menopause effects per se, but also how they interact with other variables. So whether or not it might actually not be menopause at all, it could be something that is related to menopause, as you're getting at there. To the second point, I completely agree with you. Because I show that slope for females and not males, it looks like I'm kind of suggesting that this is terrible news for everyone, who's female. I don't think that's the case. So first of all, we need longitudinal evidence. We need to this is a cross-sectional study. I would not recommend that anyone dives in and it assumes that everything is going to be terrible from midlife if you're a female. We actually think that it might just be a case.
Thank you. That's encouraging.
Yeah, exactly. But also, like more broadly, I think it's important that we look at this stuff. Partly because it's so, like I showed that figure where kind of women's health factors are so neglected in the literature. And that's both, and I don't mean that, I frame it a little bit in terms of kind of aging and Alzheimer's disease, but I think it's important to stress the point that you make that this isn't pathological. Menopause is a natural part of reproductive aging, that we're trying to understand if it has effects, but it's not a pathology. It's just important that we look at it, that we learn whether or not there are effects of it, both positive and both that are advantageous and disadvantageous that might help aid our understanding of future interventions, health outcomes that we can kind of, by looking at this, we're ultimately going to arrive at a more equitable place, hopefully. And I get that it's having a man stand up and kind of give a talk about menopause and like, by the way.
Yes. There's a tension there.
But yes, I fully agree with what you're saying. I just hope that this contributes to a better place, not a worse place.
Thank you.
Men and women have been aging for a long time.
Sure.
I guess we kind of know.
You're
You're just quantifying that way of looking at it.
I mean, one thing that we do think in the lab is that the decline for males may just happen a little later. That it might be sharper, but it might just happen a little later. So that by the time you get to later in life, that you arrive at a similar point anyway. So it might just be a timing effect. But again, that's an empirical question, something that needs to be dug into. But yeah, I should do a reasonable job of answering your questions.
See the chat has something.
Hmm.
We have a question. If menopause has effect, does andropause then? Yeah.
Maybe.
Can you define? I don't know.
You'll have to forgive me. I don't know that there's an agreement on whether or not andropause is a thing. Yes, males like females experience changes in hormones across the lifespan, day to day. That's absolutely true, and I don't want to suggest at all that I don't want to kind of support kind of sexist stereotypes. That hormones are really relevant to females. That's obviously not the case. In fact, actually, there's evidence that males show greater variability in a short period of time, but we won't go into that. In terms of, so I'm going to avoid diving into andropause per se. Is it the case that hormonal changes in males may have an effect on brain cognition? Absolutely, absolutely the case. So I would say, so I've done research in the past on testosterone, for example, in middle-aged males actually, And there does seem to be some sort of link there with cognition as well. So I don't want to kind of suggest to anyone that, you know, this is only a female issue or anything like that. That's absolutely not the case. Now, this just happens to be my area that we're focused in, the area that our lab is focusing on. But we do collect hormone measures from our males as well. So, you know, it's something that we could probe in future research for sure.
Thanks. All right, next question from the chat. It's flattery here about how interesting it is. I wonder if there's any,
The flattery.
that's why I became a scientist. I was wondering if there have been any studies or plans to do studies on including transgender patients. It'd be interesting to know how age-related cognition is affected in, say, trans men who have undergone HRT gender affirming surgeries that affect on some.
Yeah, that is such a good question. So we don't exclude trans participants from our sample. So we do, we ask a two-stage question. We ask people about their sex assigned at birth and then their current gender identity. And we also incorporate a questionnaire that aims to get at things like gender roles and things, because one of the future directions for the lab is to kind of look at those questions about whether or not some of these effects are related to what you might call biological sex, or whether or not they're more related to gender, which is kind of a social-cultural, social-psychological construct. So there's that component. So we don't exclude trans participants. If they happen to be recruited into our sample, we take them on. For this particular analysis, we did remove anyone who was taking HRT. So there's that side of things I just want to briefly mention. But in terms of future studies with trans participants, in our lab, as I say, we do have them in the sample. There aren't many, and they weren't included in this particular analysis. So it very much depends on how recruitment goes. If there's a sufficient number, we can get maybe buy-in from, particular from LGBT groups. That could be a possibility. I have seen more of this from other labs, though. So I know that there are labs working on this to see whether or not, for example, whether or not HRT use in trans individuals might impact the brain and internal cognition, things like that. But it's very much an emerging field. There's, if we think that women's health more broadly is, under researched when it comes to trans health, I mean, we're very much in the dark on that front. So, I don't know if we have active plans, but yes, it's definitely something that has a broader community we definitely should be looking at.
Great, thanks. Any questions in the room? Perfect.
I was wondering, as a lab, are you comparing MR-based markers to other imaging modalities to understand better how they correlate with those?
So when you say other imaging modalities, things like
PET?
yeah, so no. Unfortunately, I would love it if we could bring people in and have them do amyloid and tau PET scanning, things like that. Now, what I will say is we do collect blood and we do send those up for blood biomarkers. So p-tau and things like that, we do have access to. So neurofilament light, which is a marker kind of neurodegeneration, various other blood-based biomarkers. So over time, they will become available. We have to set them up in batches. So right now we can't do that analysis, but we will be able to look at kind of imaging-based markers and alongside blood-based biomarkers. And the other thing to mention as well is that with those bloods, we also look at genetic factors. So I'm particularly interested in ApoE, which is an Alzheimer's disease risk factor. And over time, the hope is that we'll be able to look at the interaction between those factors, which touches on the point in a previous question as well that, it'll allow us to look at menopause and sex effects, not just individually, but interactively with other factors that might be relevant for brain health and cognition more broadly. I hope that answers your question.
Yeah.
So how do your results compare with studies in other parts of the world?
That's a great question. So in terms of, there's not a ton of research out there on kind of looking at using the concept in the cognitive neuroscience engaging literature. There aren't a lot of studies looking at menopause and sex, so there's not a ton we can compare to. A lot of the samples tend to be conducted in North America, sometimes Europe. So the samples that we're getting are, as with the acronym weird, they're Western, they're educated, industrialized, etc. So our ability to translate these findings to other groups is a bit limited at the moment. What I would say is that our group is currently moving from McGill to Toronto Met. And as part of that, we have a big push to increase the diversity of our samples. So we can't necessarily, we won't necessarily be able to extrapolate to different countries or continents in that way, but we will hopefully have a greater diversity of people within Canada in our samples. So we can start to look at not just things like menopause, but also whether or not those menopause effects are the same for different people. So the hope is that we can move to a place where we can do those larger analyses as the samples get bigger.
Thank you. I actually had a question. Relating to the methodology of using fMRI and MRI to study episodic memory.
Yeah.
I'm not a psychologist. I don't know much about how the brain works either, but I find it surprising, or at least interesting, to think that the same part of my brain that can tell what part of the screen a picture is on is also the same part that lets me remember that this memory happened at my parents' house in Ontario versus somewhere else in the world. Is that just how the brain?
That's a great question.
Or is like that methodology can
there are a couple of things to mention. So the types of paradigms that I showed, obviously they involve us operationalizing episodic memories in a way that we might not necessarily think about them in real life. So when looking at a cartoon image on the top left of the screen, for example, and then later recalling that a few minutes later isn't necessarily the same as the example I gave earlier about your last year's birthday party. We've already got an issue of scale, things like that. So the benefits of being able to of this method is that we can kind of study it in scanner and lab. It makes it more tractable in those kind of more autobiographical methods, I would say. So that's one of the reasons why we do it. In terms of the regions that are activated, it kind of comes down to perspective. So some people will argue that there's a more modular view of the brain where certain things do certain things, and others that it's more about the connectivity. I tend to go more in the latter camp. So yes, we do see activation in certain regions, but it's not as simple as just saying they are memory regions. So it's kind of they are associated with increased activity when we recall things, but some of those regions will be involved in other aspects of cognition. So it's kind of like there's something about the neural representations, for example, of the hippocampus that means that it's important for episodic memory, but you will also see the hippocampus important for things like spatial navigation. So there's a lot of active research around that region looking at what does that, is it that there's something about the type, is it the operations that that region is performing or the representations that it houses that explains the link between it? But I don't think it's the case that we can just say these are the memory regions. They're kind of too simple. Yeah, it's too simple, I would say. Now there are obviously regions of the brain where there's more clear, for example, visual regions, more kind of basic cognitive operations where they have a more clear sense to where they respond accordingly. But the higher order cognitive functions like memory, attention, executive function, it's not quite that simple, I would say.
Right. Thank you. Right. Any other questions, either in the room or online? Everything very nicely. Okay, with that, I'd like to thank our speaker again. Thank you all for being here, and see you next time.